Okay, so I think we are all familiar with kind of the origins question. How did Earth go from being this type of planet to basically this type of planet where everywhere we look, we see life, pretty much where we have water, and some energy and et cetera. This is the big question that my group is interested in, and I think pretty much everybody in this room is interested in. Um, and because this question is so fundamental, we've been thinking about it a long time, like we as humanity. And so I just wanted to share with you this really interesting recipe for a mouse. So this is just illustrates how people used to think about how life got started. And I originally found this from Jim's book. He actually gave me this slide, Jim Cleaves. So this is Van Helmut's recipe for a mouse. I'll just read it quickly. For if you press a piece of underwear soiled with sweat together with some wheat in an open jar, after about 21 days, the odor changes, and the ferment coming out of the underwear and penetrating through the husk of the wheat change the wheat into mice, and just mice coming out of nowhere, just spontaneous generation. But what is more remarkable is that the mice of both sexes emerge from the wheat, and these mice successfully reproduce with mice born naturally. So they already knew that mice reproduce just sexually, but then they could also be produced from dirty underwear. So that's the basic recipe for how you make this mouse. Van Helmut was no slouch himself, right? He's a pioneer in gas chemistry. But this is like how people really used to think. And so, like why? And this is the idea of a spontaneous generation, but like Buenos, I'd say, was telling us the other day, life is not just a building block or arriving at a final product here at Mouse. It's really about the process, and maybe even back then they weren't even really considering life a process, and even today we still need to get better at that feat. And so, why is it so hard to actually think about life as a process? Well, chemistry is very complex. So I want to share with you this quote from my PhD advisor, Fraser Stoddart. He wrote this really interesting article. So he's not in origin of life field, but he's known for his work in synthetic or artificial molecular machines. But what both that field and origin of life field share is they're both really interested in complex systems. So what he says is time has come for us to embrace complexity, despite the fact that everyone has their own definition of it, and put much more effort into studying complex mixtures. But when it comes to creating and understanding complex networks, chemists have been conditioned by their education and training to avoid them. We have an aversion to mixtures of molecules. So that's why it's so hard, right? It's so hard to like understand life as a process, just because we're not that great at understanding complex chemistry, I would argue. Okay, so I'm not going to talk a lot about this work today, but I just wanted to illustrate how ELSI has kind of affected my way of thinking. When I first got here, we were doing a lot of radiolysis experiments. So this is our team radiolysis, and I got to work with this amazing team of people here. And really, we were interested in using gamma rays to model complex chemistry that could lead to things like maybe RNA building blocks or amino acid building blocks. We were more interested in the process of how that happens, modeling complex chemistry, rather than just getting exactly to the compounds that we wanted. So we had a productive time during this work, and we came up with a lot of papers. But one of the papers that really kind of got me thinking again about autocatalysis, right? That had this seed planted in me earlier on, but then it keeps coming up again. So this is work that was in collaboration with Zach Adam, who's now at University of Wisconsin. And so when we took a look at this radiolysis network that we're interested in, we kind of modeled it from a network theory perspective, and it became apparent that this network of potential reactions is so complicated that there is a big potential for the emergence of autocatalysis in these radiolytic networks. So this is one of the most recent papers we published in this area. But it just got me thinking, like autocatalysis, this seems really important. I've heard everybody at ELSI, especially the theorists, just constantly talk about how important this is, and so I finally wanted to start looking into studying more autocatalytic reactions myself. Okay, so anyway, we already know this is true, right? Wherever we find these major elements, an energy source, water, inhabited on the planet Earth, we can find life, basically. Okay, but when we do find life in these places, what do we find about life? What is life's characteristics? This all comes back to like understanding life as a process. Well, first of all, we have compartmentalization. All life that we know of, on Earth at least, seems to have a compartment. Here it's shown just by a phospholipid vesicle or something like that. We also see that it has genetic information storage and replication. You know, you might just summarize this as the central dogma of biology. And of course, we have metabolism that kind of makes these things work in the first place, right? It's reaction networks that afford this type of replication and reproduction. Okay, so what do all these three kind of subsystems have in common? Well, according to Tibor Ganti, who came up with the chemoton model for it's kind of like a, I would describe it as a recipe for making a very simple cell. You know, just like there was a recipe for a mouse, here's a recipe for a cell. And what he says is that without actually going into any specific chemistry, this is kind of why I like this model, and it's just a heuristic model. What you need, according to Tibor, is you got to have a membrane system, you got to have a metabolic system, and you got to have a template system. The template's like the genetic material. And they all have to be autocatalytic. So you have the genetic polymer cycle. It's autocatalytic in the sense that, you know, like DNA can kind of replicate itself. It's autocatalytic process. The membrane cycle, and then you have the metabolic cycle. So what I'm really interested in, or questions we were asking ourselves, you know, can we somehow, you know, use some sort of prebiotic chemistry or, you know, metabolic cycle that we think it could be prebiotic in the context of this chemoton model? So that's the question that I'm really kind of interested in asking myself. How can we do that? And so I just, you know, what would Jim Torr do? I don't know if anybody knows Jim Torr, but he's kind of like an open critic of the field. And he has I actually agree with a lot of his criticisms. But one of them is we tend to use in the field a lot of would have and could have happens. And so one of his criticisms that I actually agree with, if it could have happened, show me. Okay, so I'm going to try to show you how some of this stuff is possible. Okay, so what is autocatalysis in the first place? So just taking a step back, non-chemists in the audience. We've heard today that a lot of people don't even agree on the definition, or there's multiple definitions. But here's kind of a strict mathematical definition for you. You know, it's the isothermal acceleration of the rate of a chemical reaction by products of the reaction itself. Okay, so in terms of the math, don't worry, I'm not going to show a lot of math. The rate, this is just the rate of the reaction, is proportional to that starting material A, and plus FSC is just kind of side reactions, right? So it's just the more of the product you make, faster the reaction goes. That's it. Okay, so yeah, so A is the autocatalyst, C is just all other compounds reaction, FSC is other side reactions. And okay, you get this classic sort of character sigmoidal curve, so the reaction kind of starts off slow, but after you get more and more of the autocatalytic component increasing in concentration, then you get this really kind of exponential effect. And then you run out of your food right here and you go into a saturation phase. Okay, so if you see autoamplification, like this sigmoidal curve, then it's usually interpreted as a signature of autocatalysis. Okay, but why is that special? Like, who cares? One way to interpret autocatalysis is it's a kind of a means of chemical reproduction, right? Start with one molecule of A, you have some number of C molecules, and they react to make two molecules of A. So you start with one, you end up with two. That's one perspective of autocatalysis. It's not like the exclusive definition. But if you think about it, that's kind of what DNA does in the cell. You start with one DNA, for example, and you end up with two. And if you zoom out a little bit, you can see that the whole process of cellular replication can be considered as one incredibly complex set of autocatalytic reactions. You start with one, take some food, and you end up with two. So it seems to be an intrinsic process to biology, even independent of the specific chemistry. So I think that's really interesting. And moreover, you know, with autocatalysis, you can actually get some pretty complicated behavior, lifelike in quotes, like bistability. So you can actually kind of switch your reaction network between two different states, depending on an applied stimulus. You can also get this sort of oscillatory behavior. So, you know, even without enzymes or any sort of like complicated polymeric information molecule, you can still use simple molecules that, you know, could have been around in the prebiotic environment to actually get this really kind of complicated lifelike behavior. Okay, so that's why autocatalysis I think is interesting. People have probably seen this before, you know, just as a kind of illustration of what you can get with autocatalysis. This is the famous BZ reaction. You know, this just starts from very simple sort of starting material compounds, but you can still get pretty complicated behavior. Okay, so you don't really need proteins and all that stuff to get complex chemistry, lifelike chemistry. Okay, so autocatalysis in biology. So the reductive citric acid cycle here I use as kind of an example. We heard Joseph talk today about people have been, trying to do this RTCA cycle non-enzymatically for a long time, but they haven't been able to get it to go yet, at least not in an autocatalytic fashion without enzymes. So, without enzymes, it just happens to be incredibly difficult to do. And maybe one day somebody might get it to work, or find another sort of analogous reaction like what Ram has been doing. Quite amazing work showing there could be analogous reactions that are similar to something like this. Okay, but at the moment the RTCA is not autocatalytic prebiotically. Okay, so I can't use that. That would have been nice, but I can't use it for my chematon model or experiment that I want to do. So then we turn to the foremost reaction, right? Autocatalysis on the early Earth. We don't have highly evolved enzymes. So a lot of people think foremost reaction, possible on early Earth. It's been studied a lot in that context. And this is kind of a very simplified idea of what foremost reaction is. But basically you start with some formaldehyde, some glycoaldehyde, and you can end up with more glycoaldehyde plus higher order sugars on the right hand side. And you need some calcium and high pH to make the reaction happen. Okay. And was formaldehyde abundant? Well, we heard from Ueno Sensei today that from his amazing talk that seems likely. Would you agree? Yeah. So you can get it from UV photolysis using CO as an intermediate. You can get it from the famous Miller-Urey reaction, and you can get it from impact synthesis. So, seems likely would have been at least a little bit of formaldehyde on early Earth. How much, I guess, is really kind of the question. Okay. So here is the foremost reaction, and I'm just going to kind of briefly summarize the autocatalytic part here. And I know everybody's not a chemist, so I'll walk you through it. So here's the basic idea. You start with formaldehyde and you end up with a bunch of higher order sugars. So how does the autocatalysis work? So first you start with glycoaldehyde, and you do an aldol condensation with formaldehyde to give you this glyceraldehyde, 3 carbons. You have a carbonyl migration to dihydroxyacetone. You do another aldol addition to give you this ketose. That does another migration to give you the aldose. And then you can actually fragment this C4 into 2 glycoaldehyde. Okay, so just a little disclaimer for the chemists in the audience. This is known as the Breslow cycle. It is by far not the end of the story of the foremost reaction, right? This is like a super complicated reaction. But I think the Breslow cycle is good for illustrating at least the autocatalytic part, or at least one aspect of it, in order to explain what I think is going on in the rest of the talk. Okay, so what we wanted to do is, well, can we use this sort of foremost reaction as the metabolic cycle in this chematon model in such a way that we can couple this metabolic cycle with the synthesis of the precursors needed for this template cycle? So can we couple this cycle with this one using the foremost reaction as our metabolism? So that's the big question. And of course, this gets into the RNA world hypothesis. Because I'm going to use that foremost reaction to try to make precursors for ribonucleotides. Okay, so I don't think I need to explain the RNA world hypothesis to this crowd. Does this mean, though, that I'm married to the RNA world hypothesis? No, I don't. I'm not married to it. The reason I chose RNA as a target is because it's the most well-characterized prebiotic synthesis out there for ribonucleotides, I would argue. Why start with something that's difficult to synthesize when you can start with something that's already been pretty well studied about how to do it prebiotically? And so I'll just go over that briefly, the basic history of how can you make ribonucleotides prebiotically. So Orgel, tried way back in the day, 1970, to just take some D-ribose in the nucleobase and dry it down. It doesn't work. So in response, he came up with this alternative route, which involves cyanamide. So the basic idea here is you start with the C5 sugar, you react it with cyanamide, and now you have this new intermediate. And then you can cyclize again with this other molecule known as cyanoacetylene, do a dry down, and you can actually get the canonical ribonucleotide. And then fast forward about 40 years later, Powner and Sutherland reported this pretty famous synthesis for making the same cytidine. But now you can just start with glycoaldehyde and cyanamide, react that to make this intermediate, and react again with glyceraldehyde to make this same intermediate up there. Okay, a lot of chemistry, but it's kind of important to get through this slide. But anyway, yeah, you also make a bunch of stereoisomers here too, just as a disclaimer. So can we integrate like this parts of this pathway at least with the foremost reaction? Okay, so this is really work done mostly by Jeremy Tran. He's a student of mine at UNSW. And the basic hypothesis was, well, you know, if we have cyanamide, if we just take cyanamide, add it into this foremost mixture, you know, can we make this 2-aminoxazole intermediate in red for that ribonucleotide pathway, and then get to these other pathways as well? Maybe we can use that, get to the pentoses and make this intermediate, and then if by hook or crook, maybe we can get all the way to RNA one day. But first, let's just see if we can actually get to these initial intermediates. That was the idea. So what we did was we just took a mixture of, we had our foremost mixture, so 100 millimolar of formaldehyde, a little bit of glycoaldehyde to start with as our autocatalyst, and then we added in cyanamide, and then took a look at the NMR spectroscopy of that mixture after 22 minutes of heating. So this is the initial spectrum. In the aromatic region, you really don't see anything, but then after you heat, you can see pretty clearly these two resonances that are assigned to 2-aminoxazole, so this intermediate right here. And we got a 17% yield, which for prebiotic chemistry, I think that's not too bad. You know, just by not even trying to optimize this reaction at all or anything like that. Okay, so let's look at the kinetics. So classically, like we were saying before, you know, you have this sigmoidal curve for the foremost reaction. So this is just looking at how much of the formaldehyde you've consumed. Starts off really slow. You hardly consume any of it, but as the reaction goes on, you hit this exponential phase, and then you run out of formaldehyde, and the reaction just kind of comes to a stop. It's known as the yellowing point. Okay, so will the production of these nucleotide precursors follow this same sort of kinetic trajectory? That's what we're interested in next. So in this experiment, we're looking at the kinetics now. So again, we're just taking 100 millimolar of formaldehyde, some glycoaldehyde, and then different concentrations of cyanamide, and then analyzing the amount of either formaldehyde that's left over or glycoaldehyde that's in the solution using HPLC. Okay, so here is a graph where we have 0 millimolar cyanamide, nothing added, so it looks pretty similar to what we saw on the previous slide for that sigmoidal curve. But as we add more and more cyanamide, we can see that we're delaying the onset of that exponential phase, which you can see by the consumption of formaldehyde and this spike in glycoaldehyde concentration. So it actually modifies the kinetics of the foremost reaction, which we thought was pretty cool. So you modify its kinetics, but it doesn't completely shut it down, which is interesting because we're adding like a huge excess of cyanamide. It should be able to completely react and consume this glycoaldehyde autocatalyst and then just shut down the reaction, but it doesn't. And I think that's a consequence of the fact that this is an autocatalytic reaction. Okay, so you might be thinking, well, of course you're going to make this 2-aminoxazole because, you know, you're starting with glycoaldehyde and cyanamide. Of course you're going to make it, right? What we really wanted to know is this nucleotide precursor being made from the glycoaldehyde that's being produced autocatalytically? So in this experiment, we're using C13 labeled formaldehyde. And so that way we can actually determine that any of this nucleoside intermediate that we're making, if it has the C13 label on it, we know that it's coming from the autocatalysis, right? So here's the unlabeled mass. This is done with mass spectrometry. So this one is for this unlabeled species. So you have a little bit of it, grows up a little bit over time. But by far, the majority of the product that we observe has this doubly labeled 2 carbon 13s in the structure. And that's the main product that coincides with the onset of that autoamplification phase. So most of the 2-amino oxazole we're making is actually coming from glycolaldehyde generated from the foremost reaction, not the glycolaldehyde that we start with as the autocatalyst. Okay, and so what about this other intermediate right here? So again, we turn to mass spec. So in this experiment, we just kind of switch things around. Now we're using C13 labeled glycolaldehyde and just regular non-labeled carbon-12 formaldehyde. And we can see that the major product here now is the unlabeled species that again kind of tracks nicely with the onset of that exponential phase of the foremost reaction. Okay, so we're really interested in kind of arriving at a more complete picture. Again, just got to take this pathway with a grain of salt. Just kind of illustrates all the different sort of products that we have been able to observe. Whether or not these are the exact pathways that are happening, we really don't know. It's a very complicated mixture. But one of the interesting things that we found was we think actually instead of having this pathway where you go through this intermediate 2-amino oxazole directly to this 2-amino oxazole azoline, we're actually generating this compound through this kind of Orgel pathway from the pentoses. But anyway, we're really interested in actually kind of modeling this reaction to quantitatively, it's very complicated though. But that's one of the things we're working on right now. We're working with a computational chemist to like really kind of nail out some of these details. And just an analogy to the Yamanote line, right? So this, I'm not the first one to make this analogy. This kind of looks like a metabolism, right? And in the center, you have the Yamanote line. It's like a central hub of metabolism, right? So what, as to give you an intuition about why autocatalysis is interesting or special, oops. Think about this Yamanote line, right? Imagine it only had one train. And then a passenger has to like, every time it goes to a station, a passenger wants to go to some other station over here. That train has to leave. And then the track is empty and there's no more Yamanote line, right? Autocatalysis, though, every time that train goes around the track, it duplicates itself. So there's one extra train to go to different stations. And then the next time it goes around the track, duplicate again. Right? That's kind of what's going on here, I think, in this chemistry. Or that's how I like to think about it. Okay, conclusions. So the autocatalytic foremost reaction can fuel the prebiotic, maybe not, synthesis of nucleotide precursors, at least in a manner so far that could be consistent with this chemoton model shown on the right. Okay. The synthesis of these precursors is kinetically coupled with the kinetics of the foremost reaction. So again, I'm really interested in the process of how this happens rather than just making the final product. And in essence, cyanamide, that blue compound I was showing, peels off the excess glycolaldehyde, as well as other sugars in that process, but it doesn't completely inhibit the cycle, right? And that's a consequence of autocatalysis, I think. Okay, just a couple more slides. So I always like to address the challenges too, right? This by far is not a perfect system. Okay, so the foremost reaction is notoriously unselective, and we're making a lot of other compounds which I really didn't talk about because I don't have time. Are they going to be useful at all, or are they going to like be very detrimental later on when we try to extend this model? And of course, we have to use high pH. High pH is not really compatible with things like RNA. You can cleave the RNA strands, and interferes with base pairing at high pH. And under the conditions we used, I didn't talk about this directly, but we're not really even using that 2-amino oxazole efficiently. And, we'd have no idea whether or not we can even go further to the next step in that Pounder-Sutherland pathway to make this next product in that process. So there's still a lot of challenges to be done, but I think it's a really exciting, for me, it's really exciting because it's a way to really understand, how can we start thinking about lifelike processes as opposed to just making building blocks. Okay, well, since I'm running out of time, I'll just skip this, a few more slides. We'll just skip these. Go straight to, oh yeah, I do want to mention this. So at UNSW, we just started this new chemical origins of life class, and I don't want to say too much about it because I'm running out of time, but I also have a book coming out about it chemistry primer textbook that I wrote with Jim Cleaves. And it goes along really well with a chemical sort of pre-biochemistry class. And so maybe we can talk later about, combining forces with ELSI, because I know you guys have PhD program now. So it'd be good. Maybe we can use Zoom or something to have collaborations in class. Anyway, something to talk about later. Okay, so I'd like to just thank mostly, most of the work was done by Jeremy Tran here. And the radiolysis was all done by Ri-Chen Yi. Fortunately, he wasn't here today. I know he just left. He's ELSI, obviously, and but he did a lot of that work on radiolysis. And then these guys for funding. And thanks to the ELSI admin staff, especially for arranging the flights and bringing me here. And thanks for your invitation.